Imagine this: every time you sit down to eat, you're not just fueling your body, you're making a choice that could either help fight disease or feed it. Modern science is proving something remarkably: food isn't just calories. It's medicine. Real, powerful, targeted medicine. And today, I'm going to show you how to harness that power. In this video, we'll uncover 6 extraordinary foods that don't just nourish you. They help your body heal, shut down inflammation, and even cut off the blood supply to cancer cells before they can grow. These aren't exotic superfoods only found in remote jungles. They're everyday foods, backed by science, that could change the way you eat and way you live. So if you're ready to take your health into your own hands and learn how to use your fork as a weapon against disease, let's dive in. Turmeric, a golden spice commonly used in Indian and Southeast Asian cuisine, has gained global attention not only for its flavor but for its profound medicinal properties. At the heart of turmeric's power is a compound called curcumin, a bioactive polyphenol responsible for most of turmeric's health benefits. What makes curcumin particularly remarkable is its strong anti-inflammatory and antioxidant effects, which have been the subject of hundreds of scientific studies in recent decades. Chronic inflammation is a root cause behind many modern diseases, including heart disease, cancer, metabolic syndrome, Alzheimer's, and various degenerative conditions. While acute inflammation is a necessary and helpful immune response to injury or infection, chronic inflammation can quietly damage tissues and organs over time. Curcumin has shown the ability to modulate inflammation at a molecular level by blocking the activity of GNFKB, a molecule that travels into the nuclei of cells and turns on genes related to inflammation. By suppressing this key driver of inflammation, curcumin doesn't just offer symptom relief. It helps neutralize one of the core triggers of chronic disease. Beyond its anti-inflammatory power, curcumin also exhibits anti-cancer properties, particularly in its ability to interfere with the growth and spread of cancer cells. One of the key mechanisms by which tumors grow and spread is through a process called angiogenesis, the formation of new blood vessels that feed cancer cells with oxygen and nutrients. Curcumin has been found to inhibit angiogenesis, effectively starving tumors by cutting off their blood supply. It also affects multiple signaling pathways involved in cancer cell survival, proliferation, and apoptosis, programmed cell death, giving it a multi-pronged approach in the fight against cancer at the cellular level. What makes turmeric especially compelling is that it doesn't act like a single-target pharmaceutical drug. It influences multiple biological pathways simultaneously. This broad-spectrum activity is part of what makes natural compounds like curcumin so powerful in preventive medicine. In lab studies, curcumin has shown efficacy against cancers of the colon, breast, prostate, pancreas, and more. While it is not a substitute for medical treatments, its integration into a daily diet can offer support at the cellular level to reduce risk factors and improve resilience against disease. However, curcumin on its own has relatively low bioavailability, meaning it's not easily absorbed into the bloodstream when consumed in isolation. This is why traditional cooking methods often combine turmeric with black pepper, which contains piperine, a natural substance that enhances curcumin absorption by up to 2,000%. Additionally, consuming turmeric with healthy fats such as those found in olive oil or avocado can further increase absorption. This traditional knowledge, passed down through generations, is now backed by modern science, demonstrating how ancestral wisdom and cutting-edge research can work hand in hand. Turmeric can be consumed in a variety of forms: as a fresh root, a dried ground spice, or as a supplement. Many people add it to teas, golden milk, smoothies, or savory dishes. For those seeking therapeutic doses for chronic conditions, high-quality curcumin extracts in supplement form may offer a more concentrated dose. Whether used in daily meals or as a targeted intervention, turmeric stands out as a naturally in the body's defense against inflammation and disease progression. Green tea, one of the most widely consumed beverages in the world, has long been celebrated in Eastern cultures for its health-promoting properties. In recent years, modern science has begun to catch up with ancient wisdom, uncovering the powerful bioactive compounds within green tea that contribute to its remarkable health benefits. Chief among these is a catechin known as Z-epigallocatechin 3-gallate-EZG6, a potent antioxidant that plays a central role in green tea's ability to protect the body at the cellular level. EGCG has been shown to have a profound impact on inflammation and cancer biology. Chronic inflammation, as research continues to show, is a silent driver of many chronic diseases including cancer, cardiovascular disease, and autoimmune conditions. EGCG combats inflammation by inhibiting the expression of inflammatory cytokines and enzymes such as COX-2 and IL-6. It also blocks the activation of NFKB, the same pro-inflammatory molecule suppressed by curcumin. This dual anti-inflammatory and antioxidant action makes green tea a potent daily ally in the body's effort to maintain internal balance. One of the most fascinating areas of study in recent years is EGCG's ability to influence as angiogenesis, the process by which new blood vessels form. In a healthy body, angiogenesis is essential for healing and regeneration. But in cancer, this same process becomes hijacked, allowing tumors to develop their own blood supply to feed rapid growth. EGC disrupts this pathological angiogenesis by inhibiting vascular endothelial growth factor VEGF and other signaling pathways tumors use to encourage vessel growth. By cutting off this nutrient supply, EGCG may help to starve cancer cells and slow or stop their progression. Additionally, EGCG has demonstrated the ability to interfere with several phases of cancer development from initiation and promotion to progression. It influences cell signaling in a way that encourages apoptosis or programmed cell death in abnormal cells, while leaving healthy cells unharmed. Studies have also indicated that EGCG can inhibit metastasis, the process by which cancer spreads from one organ to another, by reducing the activity of enzymes that degrade extracellular matrix and support tumor invasion. Green tea also plays a protective role in the body's detoxification systems. It activates natural antioxidant enzymes like glutathione peroxidase and superoxide dismutase, which are crucial for neutralizing free radicals and supporting the liver in processing toxins. This action not only protects cells from oxidative damage but also enhances the body's ability to manage everyday environmental stressors and internal metabolic waste. For optimal benefits, green tea is best consumed freshly brewed without added sugar or dairy, which can interfere with the bioavailability of catechins. While all green tea contains beneficial compounds, varieties such as matcha, which involves consuming the whole leaf in powdered form, provide an even higher concentration of EGG and other antioxidants. Regular consumption of green tea has been associated with reduced risk of several types of cancer, including breast, prostate, and colorectal cancers, as well as improvements in metabolic health, cognitive function, and cardiovascular markers. Drinking 2 to 4 cups of green tea per day provides a steady supply of EGCG and other polyphenols that support cellular health and inflammation control. Tomatoes are one of the most commonly consumed fruits in the world, and their health benefits extend far beyond their versatility in the kitchen. Rich in a variety of nutrients including vitamins C, K, and potassium, tomatoes are especially prized for their high concentration of S-lycopene, a powerful carotenoid with potent antioxidant properties. Lycopene is responsible for the vibrant red color of ripe tomatoes and has been extensively studied for its role in protecting the body from oxidative stress and inflammation, both of which are deeply involved in the development of chronic diseases, including cancer. Oxidative stress occurs when the body's production of free radicals, unstable molecules that can damage cells, outpaces its ability to neutralize them with antioxidants. This imbalance can lead to cellular damage, DNA mutations, and chronic inflammation. Lycopene helps counteract this process by neutralizing free radicals and protecting cell membranes from oxidative harm. What sets lycopene apart from many other antioxidants is its unique molecular structure, which allows it to efficiently quench singlet oxygen, one of the most reactive and damaging forms of free radicals in the body. In addition to its antioxidant capabilities, lycopene has demonstrated anti-inflammatory effects in both laboratory and human studies. Chronic low-grade inflammation is a silent contributor to many diseases, and lycopene helps suppress the production of pro-inflammatory cytokines such as interleukin-6 and tumor necrosis factor and tumor nepha TNFF. By modulating the inflammatory response, lycopene can help maintain a more balanced immune function, reducing the risk of conditions driven by persistent inflammation. One of the most compelling areas of lycopene research is its role in cancer prevention and progression control. Lycopene has been shown to interfere with several of cancer development, including cell proliferation, apoptosis, programmed cell death, and angiogenesis. Studies have found that lycopene inhibits the growth of cancer cells in the prostate, breast, lung, and colon by disrupting signaling pathways that tumors rely on for survival and replication. In prostate cancer in particular, there is a significant body of evidence linking higher lycopene intake with reduced risk and slower progression. Lycopene's effect on angiogenesis, the formation of new blood vessels that support tumor growth, is another key reason it has gained attention in cancer research. By downregulating angiogenic factors such as VEGFs, a fuller endothelial growth factor, lycopene can potentially reduce the blood supply tumors need to grow and spread. This mechanism is particularly important because angiogenesis is essential not only for tumor expansion but also for metastasis, the spread of cancer to distant organs. The bioavailability of lycopene improves significantly when tomatoes are cooked, especially with a small amount of healthy fat such as olive oil. This makes tomato-based sauces, soups, and roasted preparations more potent sources of lycopene than raw tomatoes. Processing breaks down the tomato's cell matrix, making the lycopene more accessible to the body, and fat helps facilitate its absorption as lycopene is fat-soluble. This synergy of heat and fat enhances the health benefits of tomato-based meals, aligning traditional culinary practices with scientific insight. Tomatoes also contain other health-supporting compounds such as beta-carotene, lutein, and zeaxanthin, which contribute to eye health, immune function, and additional antioxidant protection. Broccoli, along with other cruciferous vegetables like cauliflower, kale, Brussels sprouts, and cabbage, holds a unique place in the world of nutrition due to its dense concentration of bioactive compounds that actively support the body's defense systems. One of the most potent compounds found in broccoli is sulforaphane, a sulfur-rich molecule with strong anti-inflammatory, antioxidant, and anticancer properties. Sulforaphane is not present in broccoli in its active form but is produced when the vegetable is chopped or chewed, triggering a reaction between a precursor compound called glucoraphanin and an enzyme called myrosinase. Sulforaphane works at the genetic level, activating NRF2, a transcription factor that regulates the expression of a wide array of protective antioxidant and detoxification enzymes. This pathway plays a critical role in defending cells against oxidative stress, inflammation, and chemical damage. By turning on genes that help neutralize free radicals and clear out cellular waste, sulforaphane helps reinforce the body's natural resilience to environmental toxins and metabolic stress. This molecular activation is a cornerstone of the body's ability to repair and regenerate itself, making broccoli a food with deep therapeutic potential. Inflammation, particularly when it becomes chronic and low-grade, is a driver of many degenerative diseases. Sulforaphane combats inflammation by inhibiting the expression of pro-inflammatory cytokines and enzymes like COX-2 and IL-1. In doing so, it helps restore immune balance and prevent the kind of unchecked inflammation that contributes to conditions such as arthritis, cardiovascular disease, neurodegeneration, and even cancer. What makes sulforaphane distinct is its ability to address inflammation not just symptomatically, but at its genetic root by turning off the switches that perpetuate inflammatory signals. Sulforaphane has also shown remarkable promise in cancer prevention and suppression. It works on multiple fronts by boosting the body's natural detoxification processes, enhancing DNA repair, inducing apoptosis in damaged cells, and inhibiting angiogenesis. Angiogenesis, the formation of new blood vessels, is essential for tumors to receive nutrients and grow. Sulforaphane interferes with this process by downregulating VEGF and other growth factors, effectively starving potential tumors of the resources they need to expand. This makes sulforaphane a potent tool in cutting off cancer supply lines before it gains traction in the body. In laboratory and animal studies, sulforaphane has demonstrated efficacy against a range of cancers, including those of the breast, prostate, lung, colon, and bladder. It's also shown the ability to sensitize cancer cells to chemotherapy while protecting healthy cells from oxidative damage, a dual benefit that supports its use in integrative cancer care. Its ability to cross the blood-brain barrier has opened up new areas of research into its potential role in neuroprotection and cognitive health, particularly in conditions like Alzheimer's and Parkinson's disease, where oxidative stress and inflammation are significant contributors to disease progression. For maximum benefit, broccoli is most effective when eaten raw or lightly steamed, as excessive heat can degrade myrosinase, the enzyme necessary for sulforaphane formation. Adding a small amount of raw broccoli or mustard seed powder to cooked broccoli can help restore this enzyme activity. Young broccoli sprouts contain significantly higher concentrations of glucoraphanin compared to mature broccoli and are considered one of the most concentrated natural sources of sulforaphane available. Incorporating broccoli and its cruciferous relatives into the diet several times a week can play a significant role in enhancing the body's natural ability to detoxify, defend, and regenerate at the cellular level. Berries, including blueberries, strawberries, raspberries, blackberries, and cranberries, are among the most nutrient-dense fruits available and have been the focus of extensive scientific research due to their powerful health-promoting properties. What sets berries apart is their exceptionally high content of A's antioxidants and polyphenols, compounds that help the body combat oxidative stress and inflammation. These natural phytochemicals include anthocyanins, ellagic acid, quercetin, and flavonoids, each of which contributes uniquely to cellular health and disease prevention. Oxidative stress occurs when free radicals, unstable molecules generated by metabolism, pollution, or lifestyle factors, accumulate and damage cellular components such as DNA, proteins, and lipids. The antioxidants found in berries are particularly effective at neutralizing these free radicals, thereby protecting cells and tissues from premature aging and mutation. Among these anthocyanins, which give berries their rich red, purple, and blue hues, are potent scavengers of free radicals and have been shown to cross the blood barrier, offering protection to the nervous system as well. Inflammation, especially when chronic, is a common underlying factor in conditions such as heart disease, cancer, obesity, type 2 diabetes, and neurodegenerative diseases. Berries contain anti-inflammatory compounds that help downregulate pro-inflammatory markers such as C-reactive protein S, CRP, and cytokines like TNF and IL-6. Regular consumption of berries has been linked to lower levels of systemic inflammation, improved immune response, and reduced risk of chronic illness. This effect is partly due to the way berry polyphenols interact with the gut microbiome, encouraging the growth of beneficial bacteria that help modulate immune function and inflammation. One of the most promising aspects of berries is their role in cancer prevention and support. Compounds such as ellagic acid and resveratrol have been found to interfere with multiple stages of cancer development. They inhibit tumor initiation by protecting DNA from damage, slow tumor progression by blocking angiogenesis, the formation of new blood vessels that feed cancer cells, and promote apoptosis, the natural process of programmed cell death that helps remove abnormal cells before they become dangerous. In particular, ellagic acid has shown strong effects in reducing the growth of tumors in the esophagus, colon, lung, and breast in various preclinical models. Berries also have a favorable impact on metabolic health, helping to regulate blood sugar, improve insulin sensitivity, and support weight management. This is due in part to their low glycemic load and high fiber content, which helps slow the digestion of sugars and reduce spikes in blood glucose. In addition, the polyphenols in berries have been shown to influence enzymes involved in carbohydrate metabolism and improve endothelial function, which is essential for maintaining healthy blood vessels and circulation. In the brain, berry compounds support cognitive function by reducing oxidative damage, enhancing neuronal signaling, and protecting against age-related decline. Studies on both animals and humans suggest that regular berry consumption may improve memory, delay brain aging, and reduce the risk of neurodegenerative diseases such as Alzheimer's and Parkinson's. This neuroprotective effect is particularly attributed to anthocyanins and flavonoids, which help maintain the structure and function of brain cells while promoting neurogenesis in critical areas like the hippocampus. Fresh, frozen, or freeze-dried berries can all retain their beneficial properties, and including a variety of them in the daily diet ensures a broad spectrum of phytonutrients. Whether added to smoothies, oatmeal, salads, or enjoyed on their own, berries offer a powerful, natural way to support the body's defenses against inflammation, oxidative stress, and disease progression at the cellular level. Mushrooms, particularly varieties such as shiitake, maitake, reishi, and turkey tail, have been revered for centuries in traditional medicine for their powerful healing properties. Today, modern science is uncovering the mechanisms behind these benefits, revealing that mushrooms are rich in bioactive compounds that support immune function, reduce inflammation, and play a potential role in cancer prevention control. Unlike many other plant-based foods, mushrooms contain epolisaccharides known as beta-glucans, which act as biological response modifiers and have been shown to influence the immune system in profound ways. Beta-glucans work by stimulating various components of the immune system, including macrophages, natural killer NK cells, and dendritic cells. These are frontline defenders that recognize and destroy abnormal cells, including cancer cells, before they can develop into disease. By enhancing the responsiveness and activity of these immune cells, mushrooms help the body maintain vigilance against threats while also regulating excessive immune responses that can lead to chronic inflammation. This balancing effect is critical for preventing the immune system from either underreacting or overreacting, both of which can lead to disease. In addition to immune modulation, many mushrooms possess anti-inflammatory and antioxidant properties. Chronic inflammation is a known contributor to the development and progression of many diseases, including cancer, heart disease, and autoimmune conditions. Compounds in mushrooms such as ergothioneine, a powerful antioxidant amino acid unique to fungi, help reduce oxidative stress by neutralizing free radicals and supporting cellular repair mechanisms. Ergothioneine has a protective effect on mitochondria, the energy-producing organelles of cells, which are often damaged by oxidative stress and are a key target in both aging and chronic disease processes. One of the most compelling effects of medicinal mushrooms is their ability to influence angiogenesis, the process by which tumors build new blood vessels to support their growth. Certain mushroom extracts, particularly those from reishi and turkey tail, have been shown to inhibit angiogenic signals such as vascular endothelial factor, VEF, effectively starving tumors by cutting off their nutrient supply. This anti-angiogenic action is complemented by other anti-cancer mechanisms, including the ability to promote apoptosis in cancerous cells and inhibit their proliferation. Several clinical and preclinical studies have examined the use of mushroom extracts as adjuncts to conventional cancer therapies. For instance, polysaccharide KSPK derived from the turkey tail mushroom has been used in Japan alongside chemotherapy for decades and is approved as an adjuvant treatment for various cancers. PSK has been shown to enhance survival rates in patients with gastric, colorectal, and breast cancers by boosting immune function and reducing tumor recurrence. Similarly, shiitake mushrooms contain a compound called lentinin, which has been studied for its ability to extend survival and improve quality of life in cancer patients undergoing chemotherapy. Beyond their effects on the immune system and cancer biology, mushrooms contribute to overall health through their nutrient profile. They are a natural source of vitamin D when exposed to sunlight, as well as B vitamins, selenium, copper, and other trace minerals that support cellular metabolism and antioxidant defense. The fiber and prebiotic compounds in mushrooms also nourish the gut microbiome, promoting digestive health and further influencing immune function through the gut-immune axis. Consuming a variety of mushrooms regularly, whether cooked, powdered, or in supplement form, can offer a broad spectrum of therapeutic benefits rooted in both ancient wisdom and modern scientific validation.